Now I can keep track of your comments. Okay, so today we are going to start a new chapter. And this is actually going back to the beginning of the book and talking about just like basics of how reactions happen, how enzymes work, how they help catalyze different reactions, and how macromolecules are built inside our body cells. So, it's placed in the beginning of the book, but I didn't really want to talk about it in the beginning of the semester until we had some relationship with these, cell components first. And we understood why it is important to learn about this stuff. I felt it would be better placed later after we had kind of talked about the general way the cells work and kind of, how signaling pathways work, why ATP is important, why enzymes are important. And now that we know their importance, we can kind of go back and think about how they work and how they're built. So the chapter talks about three different, kind of divided into three different parts. The first one is talking about how energy is utilized by cells and how cells are organized in their environment. The second one focuses more on free energy and catalysis by enzymes. So it's focused on enzymes in general. And then the final portion is talking about activated carriers and biosynthesis. And that's where they talk a lot about ATP and its use in metabolism in different reaction pathways as macromolecules are built and destroyed to create energy. So that's going to be our overview for that. So the whole actually last set of lectures, we're going to be focusing on three different chapters. All three of them are going to be focused on metabolism essentially and metabolic reaction processes. So if you go back and think about that pathway analysis assignment you're doing for the lab, you're looking at the pathways at a more kind of global level, right? You're looking at the overview, just different enzymes or different proteins that are involved in a process. You're not looking at a very molecular level where you look at individual steps and see how individual steps are organized and what's phosphorylating what and how that process is happening. So now in this set of lectures, we're going to start looking at that. Yes, so that we know the steps, we know this protein is getting activated and that's leading to this response. How does that get activated and exactly what happens inside? Okay, so going back to what we're going to be talking about for a lot of today's lectures is enzymes and enzyme kinetics. So enzymes are proteins typically, and they accelerate chemical reactions. And they are responsible for much of the chemical reactions that happen inside our body, simply because while those chemical reactions are favorable, they cannot normally occur at the temperature that our body is maintained at, and instead require a higher temperature input before that reaction can spontaneously occur. So enzymes kind of help provide an environment where those um, reactions can still occur at the physiological temperature. And they just speed up the rate of the cellular reaction so that we aren't all looking like sloths or acting like sloths, rather. Now, enzyme catalyzed reactions usually are never done in isolation. They are usually part of a signaling pathway or part of a metabolic pathway where there are several steps in the process, going from the first, full molecule that they are working with to the last products that they are getting. And they are usually combined in a series of catalytic reactions that all require individual enzymes to process the products at each point. These enzymes are highly specific. They can only work with that one specific substrate or a very small subset of substrates. And not based on their binding site, their active site. And we'll talk about that soon as well. So enzymes are basically what control our metabolism and all the reactions that make up our metabolism. So at the end of the day, when we look at metabolism, our metabolism is controlled by basically the rate of reactions that are happening inside our cells. And, these are all the reactions that are needed for us to survive and grow. Basically, all the reactions that we need in order to utilize energy from food, to create energy for our other reactions that are happening that are needed for us to grow and survive and reproduce. And it is a total sum of all the reactions that are happening, both catabolic and anabolic. So just a review, and this should be a review from chemistry. The catabolic reactions are going to be reactions or pathways where we are breaking down molecules while we are generating energy as a response. So when we break these bonds, we release energy that can then be utilized by other reactions in our cells. On the other hand, anabolic pathways or anabolic reactions are usually biosynthetic pathways where we are combining monomers or different molecules together to form a macromolecule or to form another product. And in the process, we are not going to be releasing, but rather requiring energy to be used. This energy that we use in anabolic pathways is the same one that was released by the catabolic pathways. So we utilize the energy that is released by catabolic reactions in our anabolic reactions. So the overall metabolism is going to be looking at the sum of these two sets of reactions. So cell metabolism at the end of the day, like I said, is a combination. I don't know why it's doing it this way. Is a combination is the sum of those catabolic and anabolic reactions. In general, you can think about the food that we take in as it is broken down. That is going to be catabolic pathways because that's going to release energy, some energy, and it's going to release some heat that can then be utilized to create other molecules inside our cells. Similarly, the food that is broken down gives rise to a lot of these building blocks that can be used as reactants within those anabolic pathways as well to make those new molecules that we need inside our cells that are different than the original ones that we started with. So this slide talked about, how living things, anytime that you have a living organism or a living thing in nature, it's usually going to be highly ordered and it's going to continue to maintain that order. But if we go back and think about the laws of physics that we look at, we know that typically things tend to go towards disorder. So how is it that living things can maintain and generate that order and maintain that within their system if overall overall all things are supposed to go towards entropy? Well, they do that by using the environment as their external, space that they can utilize to create that disorder while maintaining their own order. So reminding you of the laws of physics. The first law of physics, there are two parts: that energy cannot be created or destroyed. And however, this energy can be transferred and changed in forms. So while we cannot destroy or create external energy with extra energy inside our body, we can take it from outside and then utilize it, or we can transfer it outside and utilize it in that way as well. The second law of thermodynamics is where we talk about how entropy is continuously increases overall in a closed system, right? So overall, you would expect more and more disorder to occur over time. And no energy transfer that we create is going to be 100% efficient. So some of it will get lost in the process. However, when it gets lost, it has to get lost somewhere. And so it could be as heat that is helped to maintain our body temperatures, or it could be dissipated outside as well, released outside into the environment as well. So this, I loved the example that they showed, that a spontaneous reaction is which is going to increase disorder and entropy because it involves breaking down of the molecules, or it involves more and more disorder to be formed. It's basically breaking down those orderly molecules into smaller components. On the other hand, anytime that you're trying to organize these systems and anytime you're trying to build these molecules back up, you're going to require some energy input. So you will need actual input of energy before you can create order in the system. That order that we need to maintain as living organisms is taken, is, basically maintained through those metabolic pathways, through the heat or through the energy that is produced by the catabolic pathways inside our systems. So cells, like I said, remain orderly because that's part of living things. However, they do that, they don't defy the second law of thermodynamics. They still do that by then, as a result, doing one of two things. One, they will create energy or heat. They will release heat as they input energy into a system. And that heat, when it is released, is going to be what our body uses to maintain our body temperature, but also can be used to do work in our environmental system. And secondly, overall, what we end up doing is create entropy within the environment that we are surviving in. So while we are maintaining order inside, we are creating disorder outside. For plants, especially that are taking in energy from the environment, it works a little bit differently, right? Because they can take this, energy from the environment, from the photons of sunlight, from inorganic molecules, and they can use all that energy to create those chemical bonds needed to build the macromolecules to survive, to make the glucose, to make the actual food for themselves, so they can then break it down again and then get the energy from there. So by doing that, especially our plants, right, they are converting energy from one form to another. Again, similar to how we would look at the first law of thermodynamics. Now, when we do that in a plant setting, right, you're taking the electromagnetic light energy that is then getting utilized by chlorophyll molecule, kind of acting like a solar energy cell, to then use that energy to create those macromolecules and run photosynthesis. In our normal body systems, which are, in animal systems, you are still converting energy from one form to another all the time, right? Except usually in our case, it is going to be in the form of ATP that is then broken down for energy used to create bonds. It would be in the form of heat energy that is produced from breaking down chemical bonds. So in our case, we are using it as well to convert energy from one form to another in order to utilize it for our daily needs. So these are possible ways that you can convert energy from one way to another. Again, a lot of this should be, reviewed from physics or chemistry. Well, most of this is physics. So one of them was using our potential energy that we have inside our body, stored in our body, and convert it into kinetic energy or to heat energy. So the example of this would be if you raise a brick up, and then as the brick is thrown and falls, it is converted to kinetic energy. If it hits the floor or hits someplace, or you rub it against a surface, you in the process produce heat energy. So energy can be, again, converted to multiple different ways. The one that we use in our body the most are this type of the energy conversion that is shown in B, where you have chemical bond energy. And as those chemical bonds are broken, that energy is released in the form of heat that is then dispersed in the surroundings to maintain our metabolism. And the second one is for plants specifically, the electromagnetic light energy that is converted into chemical bond energy as you use it to build the bonds to make glucose so that you can use it for food. So this is just looking at that process a little bit more in detail, and we'll talk about this process, how these are just two flip side of the coins later on as well, that in the process of photosynthesis, in the first part, in the first step, you are capturing the sunlight or capturing the photons from the sun to create light and to use that light energy. And then we are taking in water that is then broken down to make the molecules that we need to make. In the process, as these bonds are broken down, you're releasing the oxygen, and then you are creating activated molecules of ATP and NADPH that can then take those extra energy to manufacture bonds to create your glucose molecules in that second step. So at each step, you are going to have carriers of energy in the form of carrier molecules in our body, which are your NADH, ATP, your FAD, all of those, and those will carry that energy to be used in as chemical bond energy that can then be utilized to create our sugar molecules. So at the end of the day, one of the things that we will notice about living things is that all living things require carbon in some form or another. And these carbon atoms are constantly cycling through various phases of life and various phases of energy cycle in the biosphere. So you take the carbon dioxide from atmosphere to, in the form of, again, the plants to use in the photosynthesis, so that it can be made into food. And those macromolecules all contain carbon molecules that are then broken down to create energy for the various systems that we are, various pathways that we, or reactions that are occurring inside our cells. Again, in photosynthesis, you are taking your carbon dioxide and water from the air to make oxygen and sugars, breaking it, recombining it. It's not even so much as broken down, but rather recombining it to create your glucose and oxygen that is released into the atmosphere. And in cell respiration, you take those same components that you had just made in photosynthesis, and this time you reorganize them and break them down and reorganize them to release carbon dioxide and water. So here the input was your energy of sunlight, and here you're getting the energy from the chemical bonds that you're breaking down. Any questions so far? Our students have become quieter and quieter as the semester has progressed. Ryan is back. Okay. Okay. So next we'll talk about how that energy transfer occurs. And so energy transfer a lot of time occurs through transfer of electrons. And so there are going to be basically one molecule in each case that's going to be losing the electrons, and the second one that's going to be gaining the electrons. So a lot of the reaction, the metabolic reactions in our body at the end of the day are oxidation and reduction, or involve oxidation and reductions. In the oxidation, a molecule that is getting oxidized is going to be the one that loses electrons. And in reduction, you have a gain of electron where the molecule is gaining the electrons as a result of this process. So here you have an example of two atoms that are going to form together, combine together in a polar covalent bond. When you form a polar covalent bond, one of the atoms is going to kind of hog the electrons more for a longer time than the other, maybe because of the size, usually because of a larger size, because they have more electrons, more protons, more neutrons that can pull the electrons more towards themselves. So the atom that has the partial negative charge in the final molecule is going to be the reduced atom, and the second one is the one that is getting stripped of the electrons, is going to be the oxidized side of that molecule. So in our natural reactions, like I said, we talked about how we have carbon compounds. So a lot of these carbon compounds have carbon hydrogen bonds. And so at each step of this oxidization process, one of these hydrogen is essentially getting taken and replaced with either an oxygen alone or an oxygen and hydrogen together. So here you have a simple methane molecule, considering a single carbon with 4 hydrogens. As it is oxidized more and more, each time you're essentially adding an oxygen bond to it. So in a methanol, there is a single oxygen bond. It's a hydroxyl. In formaldehyde, there are 2 oxygen, a double oxygen bond. In a formic acid, there's a double oxygen bond, and then a hydroxyl. So each time you're replacing one of the hydrogen with an oxygen bond. And in a carbon dioxide, it's going to be 2 double bonds with 2 oxygens. So reduction is essentially occurring as you are increasing the number of hydrogens in that molecule, and oxidation is occurring as you're adding more and more oxygens to that molecule and stripping away those hydrogens. So just to review this first part, we maintain biological order in our systems because we can release the extra heat into the environment and cause disorder in the environment instead of disorder inside. Cells are able to convert energy from one form to another. The most common forms of energy conversion that we have inside our cellular system, not as an organism, but as a cellular system at a molecular level, is either converting our chemical bond energy into actual heat energy, or converting our photoelectric energy or photomagnetic energy rather from sunlight into chemical bond energy. Photosynthetic organisms are the ones that use light as a way for energy input and synthesize it for organic molecule building in the form of glucose. And cells obtain energy basically by breaking down or oxidizing organic molecules. Each time as they do that, they are releasing heat energy as well as electrons that are bound to carrier molecules for energy input into other reactions. Oxidation and reduction involve electron transfers between those 2 molecules or atoms that are combining together or that are separating in the reaction together. Any questions?
Excuse me, Professor, just like I have a concern about like the drinks that they claim they are antioxidant, what they do and why they are beneficiary.
You're talking about antioxidants?
Yes, since like you just show us the oxidation process, the more oxygen bound we will have, like what this is, this something related to it, the antioxidant, or no?
Kind of, sort of. So we'll talk more about that actually maybe next week. I don't know why, in one of the other chapters, but in a not antioxidant, but in a reactive oxygen species, basically what you are getting are these, because of these oxidized and oxidation and reduction reactions, you're getting these ions essentially that are, or you're getting compounds broken down from other molecules that have highly negative charge or highly positive charge, right? And those especially those highly negative charged molecules that usually contain oxygen in them, which is what will be the basic kind of point of reactive oxygen species, are harmful to our body because they are highly, they are able to interact with a lot of different molecules in our system and break down bonds and destroy them. And
Are
so the antioxidants work by kind of destroying that negative charge, right? They work by breaking those down in a way so that they are no longer reactive, which they are in their ionic state.
oh, so are those oxidative like free? Are those like the free radicals?
Yes.
The one. Okay. Yeah. Got it now.
Okay.
Thank you.
Yeah. So usually, like these are usually coupled reactions. So you're not going to get these to be present all the time. But many of our reactions in our body, just normal living reactions, do create reactive oxygen species, some more than others. And those are the ones that you are trying to destroy by taking antioxidants. Okay.
Okay. Got it. Thank you.
Okay, so let's talk about now how enzymes specifically work. Now, I know, like, back in the day when a lot of this, again, was done back in the day when you did GB1 as well, and maybe even later. But we always talk about how enzymes help move the reaction, the rate of reaction, and make it, quicker, but in some way by lowering that activation energy. And that can sometimes make you think that, well, it's going to help a reaction proceed even if it is not favorable, right? Even if it is not going to be normally occurring. But that's actually not true. An enzyme can only help a reaction that would have occurred spontaneously had it been provided the right environment or temperature. It cannot help proceed a reaction that wouldn't have occurred spontaneously. Um, so that is an important point to remember about enzyme kinetics, that enzymes can only help those reactions that are energetically favorable, but they just needed a little push in their current setting to help them over that hump so that they could create that product. So enzymes do that by reducing the amount of activation energy that is needed by that reaction so that the reactants can be converted into products a little bit easier and at a in a more favorable manner. And for that, in order to do that, they usually need to provide those reactants or that reactant with just that right environment, where in a mini setting, which they create through their activation site, so that they can hold on to the reactant in the correct conformation, so they can provide a mini environment where the pH is lower, maybe, or the environment is favorable for that reaction to spontaneously occur and create the products. Um, so in this case, as the chemical reaction is proceeding, you will still have loss of free energy, just like you would have in any other reaction as a result of the formation of products as well. Now, enzymes have to still obey the same law of physics that normal bodies do and other atoms do. So enzymes will increase the rate of those favorable reactions, but they can only do that for spontaneous reactions. And they will still create disorder because they will be releasing free energy as a result into the environment, in this case, inside the body, to maintain the metabolic state of the body. And our enzymes cannot force a reaction that wouldn't have normally occurred given the right environment. So any reaction that requires actual energy input is a reaction that is non-spontaneous and cannot be fueled or cannot be helped by an enzyme alone. That still requires ATP input or GTP input, whatever form of energy they need in order to create that, in order to use that energy to create those bonds. So an enzyme alone won't make that reaction happen without input of energy if that was the need for that reaction. Does that make sense, guys? Yes, no?
Yes, it does. Thank you.
Okay. So what the enzyme can do is provide that environment, like I said, to lower the activation energy so that there is a higher probability that a reaction can occur in the system that they are in. So molecules generally will need you will need usually a certain number, a threshold of molecules to be there before the reaction can occur, or a certain temperature to be there before the reaction can occur. And by providing that smaller mini space for that reaction, it helps to lower that activation energy. And that allows the reaction to move a little bit easier. So in a typical setting, you will require a lot more energy input in per molecule in order for the reaction to occur. And it actually shows you right here in this graph that you require a certain amount of energy input to undergo a catalyzed reaction. However, if you have higher energy input, then you can lower that activation energy, or if you have the right environment, you can lower that activation energy so that those reactions can go and flow at a higher rate in their current setting. In the process, the enzyme itself is not changed in any way, shape, or form. So it's not like ATP, which will give off that phosphate and then be converted to ADP. Instead, the enzyme remains completely unchanged. It's only a holder, or only a providing that little room for that enzyme, for that reactant to bind, to convert into product, and then be released. And it does so by having an active site that the molecule binds to. And we've talked about how that active site helps to hold that molecule in place, and how they are able to process that system. Now, in that process, the same, again, laws of physics apply. So the free energy change will determine whether a reaction can occur or not. For a spontaneous reaction to occur, the free energy of that change of that system should be, the free energy in that system is going to be usually negative, to make sure that it is an energetically favorable reaction. So the free energy is essentially whatever energy the system has to use in order to make that reaction occur. In the case of a spontaneous reaction, you don't need input of that free energy. You're rather going to be releasing the energy back into the system. In a non-spontaneous reaction, or in an energetically unfavorable reaction, you are going to be using some of that free energy and not releasing it back into the environment, right? So in a spontaneous reaction, the change in energy is going to be less than zero. And in a non-spontaneous reaction, the change in free energy is going to be positive, because you're using more energy than you started with. In a body setting, the only way an unfavorable reaction can occur is if you have it coupled with an energetically favorable reaction, because you're going to take this energy that you take from the first one and put it into the second one in order to have that occur. Otherwise, these reactions will not happen on their own. And so typically you will notice that in your cascade of in a signaling pathway or in a metabolic pathway, you will have an enzyme coupled reaction. You will have several enzyme coupled reactions where one of them is giving off energy, and then the second process, the one that comes after it, uses that energy to create the final product. So by doing this reaction coupling, you can help to move those unfavorable reactions and have them happen inside the body, even though you cannot take in more energy from outside or raise the temperature or do any of those things that would not allow you to survive in the environmental setting. So in this case, you had one reaction, X to Y, that was driven by a positive change in energy, in free energy, because it released energy. This released energy was then used by the second reaction chain from C to D, where you had to use energy, some of this energy, in order for this reaction to occur. So this one had a change of negative delta G because you took away some of that free energy in order for this reaction to occur. So the free energy and when it's positive and when it's negative is clear.
I got a little bit confused.
Okay.
We said when it's negative, it's a spontaneously occurring one, and it's
Spontaneously. So
oh,
free energy is basically what's outside, right? So if a reaction is spontaneously occurring, it's going to throw more free energy into the system, right? Because it's going to release energy. So the change in the overall system, the delta G in the environment would be more energy. So you had 1 unit of free energy outside, a spontaneous reaction occurred and it threw another 1 unit outside. So now there are 2 units outside. So it will be a positive change in free energy, energy that is not, kind of held up in chemical bonds or in some other way.
okay, yeah, got it now.
And then when you release, when you use that energy, right, when you go through a non-spontaneous reaction, you take some of that energy in. So you took away one of those units so that you can catalyze your reaction, then that would lead to a negative change in free energy. Okay.
And they say like it's unfavorable because like our body does not like to lose energy.
Well, so unfavorable, it's not our body, but generally a reaction is unfavorable if it is going to require energy input. Nobody likes to do work, including apparently atoms. So if they have to input any work into it or any energy into a system, they don't want to do it. If it can be freebie, if it can just happen on its own, that's great. So a favorable reaction is one that can just happen spontaneously and doesn't need any energy input. If anything, it's going to give me some more energy to play with. And an unfavorable reaction is one where you require work, you require energy input, you require to put some effort before that thing can happen, and you're not getting anything back from it as such in the form of energy, right?
Yes, it's clear now. Thank you.
Okay. In our body systems, because our body is, a semi-closed system in this way, you will eventually, for every single molecule that you're, or every single chain of these reactions that you're going through, your body will have some sort of an equilibrium reached at some point where there is no net change in the, in your two products, and it's the point where the change in your free energy is essentially zero. So going from step 1 to step 4, the overall change at that point is going to be just no net change of energy. And that's going to be where chemical equilibrium is reached between those reactions. So we can look at this in equilibrium, in an equation form, where we can look at whether our energy shift is positive or negative, a change in energy is positive or negative, and relate it to whether the reaction will occur spontaneously or not, right? And so here you have these two reactions. You have the A plus B, and it's coupled with a second, C plus D products that you're getting produced as a result. So your, the free energy change is going to be the total change that you are getting between your reactants and the products that are getting produced. And that's what you're going to look at. And we'll talk about actually this equilibrium constant later on. So we'll talk about that in a minute, where it will become a little bit more clearer to you how to look at that. So the changes in free energy of your reaction are sequential, so are added. So if you have 6 reactions in a row that are all coupled to each other, The total change that you're going to look at is the change of free energy is going to be an additive effect. So you're going to take all those numbers that you get for individual change in energy, and, or rather right here, you will take the individual changes in free energy from each part of your coupled reaction chain, and you will add all of those up to get your final answer. And for an equilibrium constant to be, or equilibrium point to be occurring, all that net change should be 0. Okay. So for each mini reaction, you have an equilibrium point for that reaction. However, when they are coupled, then the equilibrium point is different, right? So individually, these may have very different equilibrium point where the change, delta change in free energy or change in free energy is 0. But when you couple them together, it's going to be related to the overall change in energy of all the various products that are there. Does that make sense, guys, or no? Yes, no?
Yes.
Okay. Okay, so let's look at an example of this one. So here we have coupled reactions, which share, coupled reactions are basically ones where you have at least one intermediate step, which is shared by both the reactions that are present within that chain of metabolic pathway. So individually, the single reactions will produce a certain amount of free energy. So let's look at the first one. In the single reaction, you have a glucose and fructose molecule coming together to give you sucrose, and the delta G is plus 23 kilojoules per mole. Is that a spontaneous or non-spontaneous reaction?
Non-spontaneous.
It is a non-spontaneous reaction. Exactly. Net result would be that this will not happen on its own, right? Because the free energy change is positive. Now, let's look at one where you have ATP and it is broken down to release the phosphate, and your net delta G or net energy change is negative 30.5 kilojoules per mole. Was that going to happen spontaneously given the right environment?
Yes, it will.
So in a coupled reaction, you would not look at these individually, but you would rather combine all those steps together and get an overall sum to get the final answer regarding whether this will happen or not. So a delta G of a coupled reaction where you have a glucose molecule that has, that gets a phosphate added to it, and then it is combined with a fructose to create sucrose. You will take the energy from the first reaction, for the single reaction, and you will add that delta G to the second reaction's delta G to get a final answer, which in this case is negative 7.5 kilojoules per mole. So would this coupled reaction work? So individually, this one would not, single reaction would not have normally occurred, right? But when you couple it with this ATP-driven hydrolysis, essentially, what happens? Does this happen now?
Yes.
Right. So it takes the energy that is gotten from all this energy that is released through hydrolysis of ATP, and that energy is then used to drive that second unfavorable reaction. So on the test, you should be able to, given a set of reactions and provided their information, given a metabolic chain and provided delta G for individual steps, you should be able to figure out if it is a favorable or an unfavorable metabolic chain. Okay. So the equilibrium constant that I showed you earlier in the equation, and we'll see that equation again later, is basically a constant that measures whether the reaction is going to be moving forward or not. So for a reaction to occur, you want the equilibrium constant around 1 so it can, or in a positive direction so it can occur, right? So in a particular setting, when the delta G is 0 and there is no net change in energy, the equilibrium constant would be 1, right? And then when the delta G changes, it's going to then move towards the positive or the above 1 or below 1. And the equilibrium constant is taken by taking the ratio of change from the products over the reactants. So here, let's look at that a little bit more in detail. You have your actual two reactants in this case that are going to combine together to make our product. So the association rate is going to be dependent upon an association rate constant, which you will know from reactions, and then from enzyme kinetics. And then you will have a certain, it will be dependent on the concentration of each one of the reactants that are present there. So the association rate is going to be a product of these three numbers. A dissociation rate is going to be basically similarly, but in this case, you will have, again, a constant for the dissociation for this particular product. And then you will look at just the concentration of the product and how easily it can get broken off. So at equilibrium, these two should be equal. That's why it's going to be your equilibrium constant would be 1 at that point. So your association rate of building those products should be equal to the dissociation rate at that point. Does that make sense? Yes, no, maybe. Okay. I have no answers, and I don't know if people are writing in the chat.
Yes.
Okay, thank you. Thank you. Okay, so here are some examples for this. So if you have 1,000 molecules of A and 1,000 molecules of B, and you're provided with the constant, you should be able to, again, calculate the overall dissociation rate or the association rate, right? So in this case, we are told that at equilibrium, you would have a certain number of your molecules, each one of your reactants, and a certain amount of product. Now, what you will notice is that just slight changes in those equilibrium constants will have a really big effect, or a slight change in concentration of your molecules will have a really big effect at the end of the day on how well they can bind and where that equilibrium constant occurs. So in the second one, it's talking about how in this case, the reaction has a weaker interaction. And so it has a lower equilibrium constant. And what you end up with is a lot more product, a lot more reactants than product. So here, this reaction was favorable. However, in the second one, the reaction doesn't look like it is that favorable at all. And it moves at a very slow rate. And so this would be an example of how the reaction works, probably under enzyme with enzyme catalysis versus an example of something that is allowed to just happen on its own. It's going to go at a much slower rate in that case. Okay.
Now it makes sense for me. I got it now when you explain the example.
Okay, I am glad. So yeah, this was an example of something different, but it also shows you how enzymes help provide that environment so that those bonds can occur better and allow that reaction to occur easier and get more product from that reaction. So in our body, in general, we have to have certain amount of thermal energy or heat energy in order for molecules to be mobile enough so that they can interact with each other and they can find their substrate and they can do the work that they need to do. If you were to lower the temperature, the metabolic rate goes down, way down, because those molecules are not as well in motion. They are not able to interact with other molecules in their space and find the substrates that they need to in order to have the reaction occur. So if you had a reaction that was only, that was possible, that was best, most efficient under 37 degrees Celsius or physiological temperature, if you move it to room temperature, you would expect that rate of reaction to go down, way down, right? Another thing is that enzymes, while they can help move the reaction rate forward, they cannot change the equilibrium point of the reaction beyond a certain point, right? So if your uncatalyzed reaction at equilibrium has a particular rate, the same rate is going to happen after enzyme catalyzed reaction as well. It's going to make that go faster, but it won't change that overall equilibrium rate. So that constant remains the same regardless of enzymes being present there. Does that make sense? Yes, no, maybe?
Yes, it does.
Okay. So the enzyme's performance basically depends on how quickly it can help the reactants get together to create the product or get in the right conformation to create the product. In general, the higher the substrate concentration, the quicker the enzyme rate, the rate of reaction will increase as the concentration of substrate increases until it reaches a point where basically all the enzyme is saturated with your substrate. And at that point, you would eventually flatten out the curve and slow down that process. So the rate of reaction for a substrate, for any reaction, as the concentration increases of the substrate, you're increasing the amount of product that is getting made, the reaction is getting made. At half of Vmax is basically the point which is reaching the maximum rate of that reaction. And that goes back to your reaction constant that is maintained regardless of whether or not the reaction is going to occur, right? Or whether or not the enzyme is there. So as you get close to that equilibrium state, that K constant, you are going to, you're going to start to slow down that rate of reaction, eventually leading to a maximum state where it can no longer affect, no matter how much more substrate you add, it won't change that rate of reaction any further. And that's the point at which it will be maximized. So the Michaelis-Menten kinetics basically look for this exact point, the amount that the Vmax where there is no where the reaction plateaus off, and no matter how much more substrate you add, you're not going to get any more product. And also it looks at the optimum point of optimum concentration of substrate where you will get the maximum effect of your rate of reaction from your enzyme. So the Km is the substrate concentration at which you get the highest rate of reaction within, or about the highest, rate of reaction in your with your enzyme kinetics. So it's half the maximum velocity, but that's the point where it's going to start to plateau off essentially. And at the high substrate concentrations, there's going to be a point where no matter how much more substrate I provided, the enzymes are saturated. They cannot take any more, and that's going to be the maximum rate of reaction for that particular compound. Or that.
Sorry, can you go back?
Yes.
I'm still trying to figure out what exactly that. So at Km, that's like, I guess, the ideal substrate concentration.
Yes. So the Michaelis-Menten constant is basically, yes, if you can think about it, the ideal substrate concentration. So until then, if you notice, it kind of increases, and the rate of reaction is increasing kind of, exponentially. It's going in a very linear way. At this point, the Km, where you reach the half of the maximum velocity, is the point where the increase starts to plateau off. So it starts to, kind of lose the same kind of momentum that it was getting. So this is the concentration that typically we run for. We look for the Km in an enzyme kinetics to look at the optimum rate of reaction within a situation for that particular set of reactions, the reactants and products. Does that make sense?
Yeah, so basically that's like the most efficient. At
Most
that
efficient,
concentration, that's the
exactly.
most efficient point.
Exactly. And after that, while it's still increasing, it's increasing at a slower rate. And then once you, there'll come a point when, as you get close to the maximum rate of reaction, that it will just plateau off. It never quite reaches that anyway. And that's the point where no matter how much more substrate you add, at that point, everything in your enzymes are saturated. They cannot function. They have they have the requirement for, they need 2 minutes to make this reaction go. There's nothing more they can do. They can't make it faster than that.
Okay, thank you.
So basically, the velocity or how long the process takes is not dependent on substrate concentration. It has to take whatever amount of time it takes. It can't do it any faster. Okay. So again, like some of you may have done these experiments in biochemistry or in GV1, where we add more and more, we add double the substrate or quadruple the substrate or half the substrate and see how the reaction moves forward. And what you will notice is that the increasing substrate concentration typically will lead to increasing products until a particular point, at which point it just plateaus off and it doesn't matter how much more you add. So the initial rate of substrate consumption, if you plot that against your substrate concentration, you can calculate the Vmax and your Michaelis-Menten constant as well. So this is the equation for that. So the velocity or the rate of your substrate consumption, how long it takes for the reaction to occur, is or you can calculate that by looking at the Vmax for that particular substrate divided by the Michaelis constant plus the concentration of substrate. So how much substrate is present, what is the most efficient point on that curve, and then that is looked at in relation to the maximum rate of reaction that can possibly happen for that particular equation. Okay? Yes, no, maybe.
Okay, sounds good.
Yes? I thought I heard a question.
No, yes, it's clear.
Okay, not you, somebody else had a question. Okay, so now enzyme activity, it doesn't mean that you cannot control enzyme activity, right? You can still control it through a few ways. You can control it partly by substrate concentration. That is one way to control the rate of reaction. You can also control it by having inhibitors of the pathway. And you can have two different types. You can have competitive inhibitors, and you can have inhibitors that are not going to bind to the exact same site. And we'll talk about a little bit of both. So if you have a substrate by itself, it's going to have the rate of reaction that it does. Typically, when you add some type of an inhibitor to that reaction, it will lower your overall rate of reaction and make it go at a slower rate, because now it will still go, but it will work a different way. Now, it does matter what type of inhibitor you have, if it is a reversible inhibitor or a, or a irreversible one that binds and never lets go. So that's going to affect this line. So sometimes it can inhibit the reaction a lot more than just a marginal inhibition. So chemical reactions will proceed in the direction where there's loss of free energy to the environment, right? So there's release of free energy, and enzymes reduce that energy needed to initiate those spontaneous reactions by providing it the right environment. The free energy change for a reaction determines whether it will occur or not. So whether the free energy is increasing is positive, which will mean a spontaneous reaction, or whether it is decreasing or getting consumed by the reaction, which would mean that it is a non-spontaneous reaction. Enzymes can only power the spontaneous reactions, and then they can use that energy in a coupled format to power the non-spontaneous reactions. Um, the free energy change always tries to proceed towards an equilibrium where the overall free energy change from the chain of reactions is 0, at which point your constant is going to be 1. So the standard free energy change is what we compare to look at the energetics of different reactions, how much total energy is getting produced or absorbed into the reaction. The equilibrium constant that we get is going to be directly proportional to delta G, or change in that free energy. So if it is spontaneous, you will see a higher equilibrium constant. Also, if you have complex reactions, the overall change of energy is additive. So you take the energy from all of them and combine it together for sequential reactions to calculate the overall change in free energy. And similarly, in those, the equilibrium constant is going to be maintained by looking at all of the reactions as a whole, not individual reactions by themselves, because those that are energetically favorable will be able to power the ones that are unfavorable typically. The equilibrium constant also indicates how strong the bindings are. So a higher equilibrium constant will indicate weaker binding, and a lower equilibrium constant will indicate quicker movement into or higher rate of reaction. And then you have many types of non-covalent interactions that allow the enzymes to bind each other to form those connections. And those are powered by just thermal energy that allows the molecules to move. So typically when you lower the temperature, you're also going to lower the rate of reactions. Any questions about this part before we go into biosynthesis? Let me see. We have about 10 minutes, so we can get through at least some of it. I know I was going a little bit faster than usual today. Yeah, we'll be able to go through most of it. So we may, like do a mini review next time before we get started on the next part. Okay, so let's talk about now how these reactions combine together to help us in our actual cell, in our metabolic pathways. So inside our bodies, many of these reactions are usually combined together in these type of metabolic chains that help us, get the energy that we need from the food as it's broken down, as well as make macromolecules that are needed to build our body systems. And so activated carriers of energy in the form of ATP, GTP, FAD, and NADH, all of those are required to take energy that is produced from catabolism of food as it's broken down, and those energy molecules are released from the chemical bond breaking, and they transfer that energy to these activated carriers that can then power those anabolic pathways or anabolic reactions, where we have these small building blocks that are then combined together to make new molecules needed for the cells. And as normally, these would be energetically unfavorable reactions and wouldn't occur without the help of this energy input. And then that leads to inactivated carrier molecules that can then go on and cycle back to get more energy from the catabolic reactions. The most common and most widely used of these activated carrier is our ATP, which is why it's called the currency of our cells. And that is basically adenine and a ribose. So it is a nucleotide that is combined with 3 phosphate groups. So it has these 2 phosphate bonds that can be broken down. And those bonds, as they are broken down, release energy that can then be used to catalyze your anabolic reactions. So in a plant, or in the case of a plant which is taking energy from sunlight, or in the case of animals which are breaking down food molecules, they are taking that energy and they are using that energy to create these ATP molecules by combining this highly energetic third phosphate on this adenine diphosphate molecule. Now, as this means that it is going to be a positive energy change because you are taking that energy in there. And then when you break it down, you will release that energy, or that will then power the anabolic reactions where energy is required as an input. Now, there are 2 things that I want to clarify here because I want you to know the difference between delta G and the standard free energy. I don't think I've clarified that well enough. So delta G is the change in free energy in the environment alone, right? And that one is going to be positive if the energy is released into the environment, and negative if the energy is consumed by the environment, right? However, the standard free energy, which is the delta G naught, the standard free energy change is the opposite of that. So the standard free energy is going to be greater than 0 or a positive as you build the bonds, and negative as you release the bonds. So that's looking at the actual reaction side. So the delta G itself looks at the free energy inside in the environment, and the delta G naught, you can think about that as the energy that is inside the reaction or inside the reactants and products themselves. Does that clarify that difference between delta G and delta G naught?
It does, but like delta G naught, if it's like bigger than 0, it's like favorable at that time when like the binding happening between the phosphates.
Yeah, because now there is more energy inside the molecule than before. So it's the same idea, but delta G is looking at free energy that is in the environment, and delta G naught is looking at the standard change in energy, which is inside the pathway or the actual reactants and products.
So like the cell will be happier to have more energy inside at this time.
Well, it's happier. Say it again.
I meant like the cell, that process is more favorable, like to build the ATP, because it will be more energy with delta G naught.
No. See, so the thing is, this requires energy input. So it's still an unfavorable reaction overall, right? It's an unfavorable reaction because delta G is below 0. It's negative. It required energy in the form of a phosphate taken in from the food energy or from sunlight to be input inside.
Okay, I see. Yes.
Right. So it's just talking about the state of the actual reactants versus state of the environment. Okay.
Yes, got it. Thank you.
Okay. So yeah, so this one we did. So ATP hydrolysis is often tied to this portion because otherwise this won't happen, right? So you have to tie it with another kind of reaction. either taking in energy from the sunlight to make ATP, or taking in energy from food to prepare ATP. And then that ATP, as it is broken down, is going to release a lot of energy that is then tied to either phosphorylation of another product, because you take that phosphate group and you put it on something else, right? To move another reaction forward in that way, whether it is a signaling pathway to activate a protein, or whether it is a metabolic reaction to help create a new molecule that can then be broken down for more energy or for use in a different system. So this third bond is a phosphoester bond, right? In this case, when it's broken down, because it's usually combined to a carbon molecule or to a another organic molecule, as it is moved over from the ATP, where it was combined to a oxygen from the second phosphate, right? So energetically unfavorable reactions are, that's how they're driven. They're driven by this ATP hydrolysis, where the ATP is converted to ADP. That phosphate that is taken off is then used on your reactants or on your products to create the new step. And so the activation step is where the phosphate is added and then to your reactant. And then the condensation step is where then that activated reactant is then combined with whatever else it needs to be, the next macromolecule, the next monomer, to create your product. And in the process, that phosphate is then let go. And usually it leads to some condensation step as an intermediate. So the activation is taking the ATP, changing it to ADP, and creating an intermediate product from your reactant that is then used to create the final product in the second step. Okay? Yes? I can't tell.
Yes.
Thank you. I like to have some type of answer from you guys, given that I can't really see you. Other carrier molecules that can also carry energy are NADH and NADPH, and these are going to be carriers for electrons and not necessarily phosphate the same way that we have ATP, right? So you have NADPH, which is a reduced electron carrier, and NADP plus, which is an oxidized electron carrier. So both of them can be electron carrier, but in different ways. NADH is used in a lot of catabolic reactions, and NADPH, on the other hand, is used when you're building molecules. So it's one of those intermediate steps where you are using it as an intermediary to build macromolecules in our system, including as we build, for example, DNA or another macromolecule. Okay. So NADPH is an activated carrier of electrons. It goes through oxidation reduction process to make NADP plus, which is the oxidized electron carrier. And then when it is reduced, it turns to NADPH. Acetyl Coa is another activated carrier. In this case, again, if you look at this molecule, it is overall a very stable molecule, but it has this carboxyl group at, or rather an acetyl group, sorry, at the end of its chain that's bound to a sulfur that is a very high energy bond. And that bond, when broken, can provide a lot of energy for use in macromolecule building. Synthesis of any type of macromolecule, whether it is a polysaccharide chain or our DNA and RNA or proteins, all of those require a lot of energy input. And those are usually fueled through one of these methods. And a lot of them are fueled through energy from some type of nucleoside triphosphate hydrolysis. Many of these bonds, if you remember, actually not just include a single bond getting broken, but two bonds two phosphate groups getting broken from that triphosphate chain hydrolysis, either GTP or ATP, so that you get AMP molecules at the end of the day to make those bonds to create the final macromolecule chains that we get. So here is an example of exactly that. In when we are building many of these macromolecules, what you get in ATP hydrolysis is not just a single phosphate group getting taken off, but rather a pyrophosphate formation where two phosphates are broken down together, and then the two phosphates are then broken down further to even create more energy production. And so both of these require a water molecule to break down first the bond between the 2-phosphate on one side and the adenosine monophosphate on the other. And then this pyrophosphate molecule is further broken down by breaking that the oxygen bond between them again within second water molecule. And so this leads to release of two phosphate molecules that can then be used to fuel reactions. So this is something that we've seen in synthesis of nucleic acid, as well as when we made RNA molecules, when we were transcribing and translating DNA, that these required that pyrophosphate, not just a monophosphate, but a pyrophosphate from the ATP. And that pyrophosphate was then used to fuel the extension of those DNA and mRNA chains. So nucleic acid is a multi-step process driven by this type of ATP hydrolysis, and with the high energy need that it has, it uses a lot of energy. A lot of ATP is used in a single round of transcription or translation of individual mRNA molecules. So the activated carriers and biosynthesis, here's a review of that. The formation of an activated carrier is usually coupled to energetically favorable reactions, so that these activated carriers can take the electrons or phosphates or energy from these molecules that we are breaking down and releasing energy from. ATP is the most widely used activated carrier that is broken down to release phosphates, and those ATP phosphates are then used to join molecules together or to activate them. Other forms of carriers include NADH and NADPH. Those two are both carriers of electrons and are used to fuel oxidation and reduction reactions, not phosphate transfers. NADPH and NADH have very different roles in the cells, and we'll talk more about them in the next chapter. Cells also make use of many other types of activated carriers, including acetyl Coa and other molecules. And the synthesis of biological polymers like nucleic acids and polysaccharides and fatty acid chains require an input of energy that is taken from hydrolysis of ATP to create pyrophosphate in many ways, which is two phosphates coming off of it instead of one, leaving behind an AMP or adenosine monophosphate. And then that is those two phosphates in the form of pyrophosphate are further broken down for more energy release. So that was chapter 3. Now next week, we are going to start to look at how cells obtain energy from food. And we will be looking at breakdown on utilization of sugars and fats and how they are used to regulate our metabolism. So that's all for this week.